Okay, so now we are going to move to a different format of talks. So I will just to thank the first three speakers, and now we are going to move to a flash talks. I will just remind people that is in the streaming that if they want to make questions, just write this. I hope that this is working. Hopefully it's working, but no questions. Okay, so we move to the first flash talk. Is a talk, 8 minutes, 2 minutes questions, okay? So the first speaker is Yasmina Manso. She's coming from, let me see, sorry. Okay, now. She's coming from the Developmental, Neurobiology and Regeneration group, and she's going to talk about spatial and cell-specific contribution of Relin expressed by Cajal-Retzius cells or GABAergic interneurons to cortical lamination. So, thank you.
Thank you very much. Good morning, everyone. First, I would like to thank the Institute of Neuroscience for the opportunity of presenting you our latest work. And first of all, I would like to introduce you to Relin. Relin is a large extracellular matrix glycoprotein, which crucial roles both during brain development and also in the adult brain. During embryonic development, Relin is mainly expressed by Cajal-Retzius cells, which are an early pioneer neuronal population which expresses calretinin, and it's located in the marginal zone of the developing cortex. At these stages, Relin has been found to be fundamental for neuronal migration, layering of laminated structures, and also synaptogenesis. However, Cajal-Retzius cells disappear by cell death within the first postnatal weeks, and at postnatal and adult stages, the main source of Relin are GABAergic interneurons, which, as you can see here in this image in green, are found scattered through all the cortical layers, including layer 1. At these stages, Relin has been involved in adult plasticity, as well as in the pathogenesis of neurodevelopmental and neuropsychiatric disorders. However, the specific contribution of Relin from GABAergic interneurons to cortical development is poorly understood. The absence of Relin during development leads to a very distinctive phenotype known as Reeler. Reeler mice are characterized by dramatic lamination alterations that include inversion of cortical layering, invasion of layer 1, or failure in the formation of the dentagyrus blades. To further understand the role of Relin in cortical lamination, we developed two transgenic lines in which the exon 1 of the Relin gene is inactivated either in calretinin-expressing cells, such as Cajal-Retzius cells, or in GAT2 expressing cells, such as GABAergic interneurons. And our objective was to understand the specific contribution of Relin from each of these cellular sources to cortical lamination. So first of all, we established the expression pattern in the Cajal-Retzius model, and we observed that Relin was absent from calretinin-positive Cajal-Retzius cells located in the marginal zone at early developmental stages. At later stages, Relin was only expressed by calretinin-negative cells that were found scattered through all the cortical layers and that were presumably GABAergic interneurons. To study cortical lamination, we used two layer-specific antibodies that concentrate either in the upper layers, such as COOX1, or in the lower layers, such as CETIP2, and that usually form two defined bands in the cortical plate. In the absence of Relin from Cajal-Retzius cells, we observed that the distribution of these layer-specific markers was wider than that of controls. Moreover, we also observed clusters of COOX1 positive cells that remained ectopically located in lower layers. In addition, we also observed a transient invasion of COOX1 positive cells into the marginal zone at E16. However, this invasion was corrected at later stages, coinciding with the onset of Relin expression by GABAergic interneurons, and suggesting that perhaps a source of Relin around this region, it's important to prevent its invasion. At later stages, the cortical defects persisted despite the abundant Relin expression by GABAergic interneurons. Using BRDU birth dating studies, we proved that these COOX1 positive cells ectopically located in lower layers were indeed upper layer fated neurons. To substantiate these findings, we also performed in utero electroporation experiments in which we injected DNA encoding GFP into the ventricle of E14.5 embryos with the aim of labeling upper layer fated neurons. When we analyzed this at P1, we observed that in line with our previous results, the absence of Relin from Cajal-Retzius cells made that many of these upper layer fated neurons failed to properly migrate and remained ectopically located in the lower layers. Moreover, many of these GFP-positive cells presented marked morphological alterations that included oblique, horizontal, or even basal orientations of the apical processes that were reminiscent of the Reelin phenotype. So next, when we characterized the GABAergic interneuron model, we observed, as expected, that Relin was expressed by Cajal-Retzius cells in the marginal zone at early developmental stages. However, at later stages, when the main source of Relin are GABAergic interneurons, Relin was virtually absent from the cortex. The lack of Relin from GABAergic interneurons led to a progressive but massive invasion of COOX1 positive cells into the upper layers of the cortex. And this phenotype of invasion persisted over time, again suggesting that perhaps we need a source of Relin permanently around this region to prevent its invasion. To test this, we performed transplantation experiments in which we transplanted GABAergic interneuron precursors tagged with GFP into the upper cortical layers of pups deficient for Relin in GABAergic interneurons. And we analyzed the invasion phenotype in these animals 2 weeks later. The transplanted mice presented numerous GFP-positive cells that integrated and migrated in the host cortex. And many of these cells, or some of these cells, also expressed Relin. Occasionally, these cells accumulated around layer 1 and even formed clusters just above this region. Interestingly, when we analyzed the invasion phenotype just beneath those clusters, we observed that there was a reduction in the number of COOX1 positive cells invading this region, in sharp contrast with the neighboring cortical areas or with the contralateral cortex. Again, suggesting that perhaps a source of Relin is important to prevent layer 1 invasion. So in conclusion, our work supports that Relin expressed by Cajal-Retzius cells and GABAergic interneurons cooperate to orchestrate neuronal migration. Moreover, our work also highlights specific roles for Relin expressed by GABAergic interneurons. For instance, the cortical alterations that we observed in the absence of Relin from Cajal-Retzius cells are not as severe as those observed in the Reelin phenotype, and perhaps suggest that Relin from GABAergic interneurons is able to compensate this phenotype partially. Moreover, our work also supports the idea that the sequential expression of Relin first by Cajal-Retzius cells and later on by GABAergic interneurons is key to prevent layer 1 invasion. I would like to thank all the people that make this work possible, and I would also like to thank you for your attention.
Thanks. Okay, so thank you. Time for questions. Okay, so I have a question, meanwhile. So it is known how this Relin secreted by the GABAergic neurons can block the invasion of this layer 1? Any idea about the mechanism?
We still don't know. We just think that it's important to have a source in that region. And it might be working as a stop signal, so a stop migrating signal. So we think it's key that there's some source of Relin, either from Cajal-Retzius cells or GABAergic interneurons, that prevents this invasion. And that's why we wanted to do the transplantation experiments, to show that when we express Relin from either source, in that region, we are able to at least partially prevent the invasion of the cells in that area and correct the over-migration deficits that we think we are seeing.
Because do you think that it's really important, the sequence, meaning that it's important that first the Cajal-Retzius cells express Relin and then later on will be the GABA ones? I mean, this temporal sequencing, do you think that it's critical?
I don't think that it matters who is the source.
Okay.
I just think that it's important that there's a source.
Okay.
So I think it doesn't matter if Relin comes from one source or the other. For instance, we have data also in the hippocampus where we see that Relin from GABAergic interneurons is mostly compensating most of the defects that we see in the Reelin are not seen in our models because Relin from both sources is able to compensate for it. But there are some specific features, for example, the formation of the infrapyramidal blade. It's missing in our Cajal-Retzius model, despite there's Relin from the GABAergic neurons. But it's probably because the location of the Cajal-Retzius cells in the fissure, it's key to provide the source of Relin for the granular cells forming the dentate gyrus. Okay. So it's location, but I don't think the source matters that much.
Okay, thanks. Questions? No questions? Okay. Oh, see, questions, sorry. How is so there's two questions from the chat. The first one is saying, in my opinion, it would be better to opt for both pillars from the beginning. It is feasible. Pillars, so the word is pillars, and also I don't understand what means pillars here in this question. So I will just, I don't know, ask the guy that just write this question if they can just clarify. But meanwhile, I go to the second one. The question is, how is the layering in the hippocampus?
I don't know if I can have the presentation back to show it because I have a slide.
It's possible? It's possible to have the, no, we cannot back to some slides? Yes, okay, we can.
So we just did the same in the hippocampus, but for a matter of time, I didn't present it. But it's more or less what I told you. In the hippocampus, we have both sources of Relin coexist. GABAergic and Cajal-Retzius and Cajal-Retzius cells express Relin even in the adulthood. And in both of our models, we see that the formation of the CA1 grossly, it's normal in both of the models. So it's meaning that either Relin is compensating for the lack of the other Relin. But as I was telling you, what we see is, as you see here, that in the infrapyramidal blade of the dentate gyrus fails to form in the absence of Relin from Cajal-Retzius cells. So it's a specific feature of the Relin expressed by Cajal-Retzius cells that GABAergic interneurons are not able to compensate in this case. And I think it's a matter of where they are located, probably. Yeah.
Next one. Okay, so the next speaker is Natàlia Gorina-Careta. She's coming from the Brainlab group, Sant Joan de Déu Institute of Neuroscience, and she's going to talk about the effects of a bilingual fetal acoustic environment on the neuronal sound encoding of newborns. Thank you.
Hello, everyone. Thank you all for being here. First of all, I'm going to talk about a little bit following up Dr. Perez talked about before how newborns encode sound at birth. So first of all, and to start with, the first concept we need to get on is what is encoding the acoustic environment. So since the moment we are born, before we are born, the babies are safe inside the mother's womb, but the moment they are born, they are exposed to a world of sounds they've never heard before. So from the moment of birth, they need to start doing the computational processing of encoding the acoustic environment, which goes from, did I hear a sound Where does it come from? What does it tell me? And do I need to do something to respond to that in adult life? So it has been demonstrated that language experience shapes how the auditory system processes sounds from the very first moments in life. And actually, it has been also shown that early childhood is the developmental window with maximal influence on neural function. So exposure to a specific language environment during this early childhood alters infant speech perception during the first year of life, even though at the moment of birth, the babies are not biased, or baby's auditory system is not biased to the native language of their parents. But the question we have here, or the question we're wondering is, what happens with learning a second language? Actually, here we are really used to speaking Catalan and Spanish simultaneously. So the question that arises is, what happens when we learn a second language? And it has been observed that learning a second language during early childhood, during this window, is a driving factor for functional neuroplasticity. And in adults, it has been observed that people who are bilingual have enhanced responses to sounds, also have enhanced sustained selective attention, auditory sustained selective attention, but also visual selective attention. And actually, it has been observed that the years of bilingualism of a person correlate with the neuronal firing consistency of the auditory system. And as Dr. Perez said before, there's an important time window which we need to consider, which is fetal hearing. So it's what happened during the neonatal period of everyone's life. So also, previous studies demonstrated that fetal hearing experience shape linguistic and musical preference of neonates from the very first moment of birth. So the aim of our project and the aim of our research line is to understand how determinant is this fetal acoustic environment for the newborn's ability to encode and process sounds, both linguistically and both with the music. This study specifically aims to analyze whether these bilingual environments, or whether if the mother that's pregnant speaks both languages, modulates the newborn's ability to encode and process sounds at the moment of birth, so before he has been exposed to any kind of auditory environment. To analyze this, we use what's called the frequency following response, which, in complicated words, is a sustained auditory evoked potential elicited to complex sounds that synchronizes to periodic components of the signal. In easy words, basically, it's a response of the neurons that actually mimics the incoming sound. So in gray here, what you see is the stimulus, and in black is the response of the neurons. So if we see that if we change the stimulus from a cello note to a piano melody, the response of the neurons changes, mimicking the incoming signal. And to test or to show you, I'm going to play some sound. I hope it plays. And I'm going to ask you if this is ascending or descending. So I would like to raise your hand if this you think is this ascending. Great. So what I'm going to tell you is what you just heard right now is the frequency following response. So it's the neural response of a person that's on the back part of the place. And actually, these are shepherd tones, and the actual sound is this one. So what you've been able to clearly identify if it was going up or down is this response that we're analyzing. So by analyzing this response, we recruit the mothers where they are pregnant at 32 weeks of gestational age. And we, during this last 3 months of pregnancy, we follow their music exposure. And then at 0 months, when the baby is born, we do an electroencephalogram and we record this frequency following response. We do some sociodemographic questionnaires, and then at 6 months, again, we retest the baby and we do some questionnaire to analyze how their language exposure has been during this first 6 months of life. For this presentation, I'm going to focus on the 0 months. So basically, we had 90 newborns, 45 of them were bilingually exposed during pregnancy, and 45 of them were monolingually exposed. And basically, we presented an OA stimulus, which is the one that we use to obtain this frequency following response, 4,000 times, whilst the day after they were born, whilst they were sleeping in the crib next to the parents' room in the maternity ward. And before going on to the results, there's one concept that's really important, is that what is the sound frequency, or what does the fundamental frequency of a sound mean? So basically, the sound frequency is the number of cycles per second that changes in pressure repeat. And it's the physical measure that we measure that is associated with our perception of pitch. So higher frequencies are associated with higher pitches, and lower frequencies are associated with lower pitches. So when recording the frequency following response, and I'm going to go directly to the important results, what we observe over here is that whilst monolingually and bilingually exposed children have the same response to the incoming sound, bilingually exposed children, which is the red line over here, are not only tracking a specific frequency of the incoming sound, but are also tracking other frequencies that are surrounding this. So basically what we observed is that this fetal monolingual environment provides a more stable background for newborns to encode and process sounds. They are more sure of the sound that they are encoding. However, the bilingually exposed newborn are tracking a wider range of frequencies. And we hypothesize that is because whilst if I'm speaking English, I have the same pitch of my voice. Si cambio a castellano, poco, pero mi tono de voz cambia. So basically, what we hypothesize is that babies who are exposed to a bilingual environment, they are tracking a wider range of frequencies to try to identify these small changes in the fundamental frequency of the sounds that are encoding, which is what they have been exposed in the belly while their mother was speaking the two different languages. So to conclude, basically, I'm going to say that prenatal language exposure actually modulates the neural responses to human speech at birth, so that at birth, for the moment, monolingual fetal exposure provides a more stable background for newborns to encode and process sounds. And actually, this points for us to hypothesize that these bilingual infants commence the process of language acquisition by separating the two languages from birth. So already from birth, they are looking into the stimulus that they are receiving, the different pitches to encode the two different languages separately. So additionally, and I'm going to do a spoiler, which is following what Dr. Perez presented before, similar effects are observed with fetal music exposure, but I'm not going to explain anything, and I'm going to ask you to come to see me in the poster session later. So I need to thank the Brainlab Cognitive Neuroscience Research Group, and also the people from Hospital Sant Joan de Déu, and especially the Baby Lab, which are the ones that have been a long time and a lot of hours with me in the hospital. And of course, thank you to all the babies and families who, if you have a healthy pregnancy, if you have a healthy baby, it needs to be thanked that they participate in studies. And thanks for all of you for listening.
Okay, so time for questions. One question at the back of the room.
So I have a very easy and fast question. All the babies that you tested, are they all bilingual in the sense of Catalan and Castellan?
Good question.
90% of them, yes, at the moment.
So you also have other babies that were, I don't know, English and Castellano, for example?
We have English, rather than Catalan and Spanish, we have English and Spanish, and we have Arab and Spanish.
Thanks.
More questions? I know that I'm running late, but I mean, we have a lot of time for lunch, so I will just take 2 more minutes. Okay, it's a short question. It's just a curiosity. So sometimes at home, I mean, when you are talking 2 different languages, maybe the proportion of the use of this language is different, meaning that one time more Catalan or Spanish or whatever. So probably it's an important point now. I mean, you take in account this proportion of?
For this study, no, but the answer is not yet. Okay. So we're doing the follow-up of the babies at 6 months, and during this 0 to 6 month period, we are actually taking into account all the languages they've been exposed and the percentage of how much exposed they've been to the different languages. So this would be the second step to check what would happen at 6 months. And the next step on the next study would be to do a more systematic approach to trying to understand how both music and language affects this prenatal exposure. The thing is that with music, we've obtained really, really nice results. So we are going to do this mixed approach to try to see if music could compensate for some other deficits, on, like prenatally and on the fetal life.
Okay,
Well,
I will visit your poster. Okay, thank you. Okay, we move to the last one, to the last talk today. Okay, so the next speaker is Anna-Christina Häb, and she's coming from the neuronal stem cells and brain damage group, and she's going to talk about human iPS-derived neurons allow dynamic monitorization of complex network activity for machine learning. Thank you, Anna.
Thank you very much. So, hello everyone. First of all, I would like to thank the Institute of Neuroscience for giving me the opportunity to introduce you to my PhD project, which is part of the NeuChip Consortium, where we want to design human iPS-derived neural cultures for machine learning. No? Okay. Yes. Okay. And as you may know, machine learning has a subfield called deep learning, which consists of complex algorithms which are inspired by the human brain structure and function, also called artificial neural networks. These have some limitations, such as ineffective learning roles or long training times and much more. And even the most impressive supercomputer is not as efficient as the human brain. So, and that's exactly the question. Why not to try to overcome or to help these limitations by implementing a real biological neural network to this system? And this is what we are aiming to do in the NeuChip project. We're aiming to generate a chip where we plate human iPS-derived cells which can perform data-driven tasks in an efficient way and have the capability to learn over time, which should, in the final, look like this, where we give some different kind of inputs, where the cells plated here in our modules, processing and then performing a task. So in order to build this chip, what would we need? We would need a biological neural network, which we need to combine the electric circuit, for example, with the help of a chip using as a neural interface. Furthermore, we want to design a neural circuit by having input areas, processing areas, and output areas, with the cell culture, which could also be trained. Starting with the first part, I want to introduce you to our cell culture part, where we use human iPS-derived long-term neural epithelial-like stem cells, which you can see here express typical neural stem cell markers such as nestin and SOX2. By using a defined protocol, we are able to generate cortical excitatory neurons, where you can see here also express typical neural markers such as MAP2 and beta-3-tubulin, as well KGA, which is a glutamase marker for excitatory neurons. Also, at day 30 of differentiation, there are some astrocytes appearing in our cell culture. You can see here in this GFAP positive staining. If you're interested in more details of these protocols, we have also a poster, number 5, talking about more about how to generate these cells. And by using lentiviral vector, which was generated with the help of Cajal-Retzius, we are able to stably express genetically encoded calcium indicators in our cell culture, which we use to measure the intercellular calcium levels in the whole network to perform indirect measurements of the indirect neural activity of our culture. But not only using this method, we are able also to use the multi-electrode array, where we perform electrophysiological recordings of our cell culture over time, where we can plate cells in this grow area here, which contains around 4,096 electrodes, which measure or which are able to measure the membrane field potential of our network. Where you can see here, we have an example of a recording in a high temporal resolution, you can see here, where we have all 4,000 electrodes and 4 different examples of a raw trace. So performing these methods or recording these electrophysiological recordings, we are also able, with the help of our NeuChip partner, Jordi Soriano's working group, perform bioinformatical data analysis, for example, here with the calcium data. And I just want to mention that we are able to perform a lot of analysis of this part, so I just give you a short introduction, just some examples which we are able to do. And for example, you can see here in this first version that we can see where the activity in our network was initiated. But also, we are able to show the row traces as well as the raster plot. In the raster plot, you can see in every row one cell line, and each dot is one neural activity. And as you can see here, that also over time, our neural network is getting stronger and also more synchronous, meaning the functional network activity. Furthermore, we are also able to analyze the connectivity, the functional connectivity within our network, and as you also can see here, that the functional connectivity is increasing over time, as you can see also, which fits together with the synchronicity since the network is getting more synchronous over time too. Furthermore, we are also able to analyze the modularity between the networks, meaning how much, or how, if the cells forming communities which are more tend to connect within each other and talk more to each other. With recently performed data analysis, I'm sorry about this. And with the data analysis, we also can see that in the first MIA recordings, we also are able to generate these results, which are similar to the calcium data. So just summing up, that we are able, with strong bioinformatical tools, with the help of Jordi working group, especially Ake Hauben, his PhD, we're able to perform a strong data analysis of the functional network activity over time of our network. So another partner of our NeuChip project is Dr. Paul Roach, a bioengineer in Loughborough University, who is designing these patternings where we have an input area, processing areas, and output areas, where we want to aim to plate our neuronal cultures with the final aim having different kind of neurons, for example, excitatory as well as inhibitor neurons. In our first trials, we can show that we are able to generate these microfluidics devices where we're able to plate already our cell culture inside. And what we are recently working right now is also to implement the final goal is to bring this patterning in our MIA chip. And this MIA chip is not only able to perform electrophysiological recordings, but also this MIA chip is able to perform precise microelectronic stimulations. And just with the first trials, we performed different kinds of stimulations in these in the node chips, where we're stimulating some of the electrodes. In our case, in this area here, so we can see here all the 4,000 electrodes, and we were stimulating this column of electrodes. And as you can see here in the raster plot in the first result, that at the beginning we have a spontaneous synchronous activity in our culture, and by introducing a stimulus, which you can see here, is that we are able to provoke in some of the measured neurons, and we are able to provoke stimulations or a reaction, functional electric reaction activity in our culture, as you can also see here in the first this diagram. And what we want to do with the help of this setup and the strong data analysis I showed you before, we want to determinize and analyze the parameters which govern this neural function, our neural network, to have this outcome. And by learning as much as possible about the different kind of stimulations and getting a different kind of output in our network, we are trying to learn as much as possible in our neural dynamics. So then, if we know which kind of stimulation we give to our network and getting this output, we want to learn, we want to reach a different kind. We want to be able to train our neural network in this precise, performing task. So where we want to fit together everything in this kind of chip, where we plate our designed neural networks, where we perform different simulations and are able to train our circuits with having a desired processing capabilities. And since I was also talking about the NeuChip Consortium, I would like to introduce you shortly to our partners. So we are stem cell biologists, neurophysicists, and bioengineers in UK and as well here in Barcelona. We have the 3Brain company, which is providing us with the MIA system, as well as computational neuroscientists in Paris and biomedical microelectronics engineers in Haifa. And in the end, I would like to thank my supervisor, Daniel Tonero, and the whole lab for taking me in this great project, as well as Jordi Soriano's working group with huge help in the data analysis, and as well as the whole laboratory, as Josep Canals in the whole in the laboratory, and as well the laboratory in Sweden for providing us with the LTNS cell lines. And thank you very much. If you have any questions.
Okay, so time for questions. So, one question.
Hello. Hello.
Oh, sorry. So there's one question. Sorry.
Don't worry. I think to understand that these systems, these neural networks, are becoming very efficient at performing a task. My question is, how plastic are they? I mean, a trained system can then be untrained or to perform another task, or you never did?
So right now we're just starting with the stimulation, so we're setting up different kind of stimulation parts. So I don't, in our cultures I cannot tell, but I think depending on how you teach the system and when you start to learn or train, perform stimulations, you can, I think, also remodel the network, which is going to be the aim of designing this chip, because in the end we want to design a chip which is not able to perform only one task, but it can be trained for giving different kind of tasks with this kind of the property of also our brain and neural cultures, that we can have different kind of tasks and we can solve them. Because right now in machine learning, for example, we train the system, but you don't provide any other information, so the system is only as good as you teach with the data you provide. And the aim is going to be more to provide also new incomes, even if it is a system it doesn't know, to be able to process these. So I think the aim is going to be to exactly target this.
Hi, very nice talk. So my question is related about how to analyze the neuronal network. No? So you are just playing with calcium imaging and MIAs. So in your hands, which is the best method to analyze the neuronal network? Using electrophysiology, using MIAs, or calcium imaging?
So I think, in my opinion, the combination of both is really good, because with the calcium you can see the cells firing, but you use it as an indirect indicator for the neural activity, while in the MIAs you not directly see the cells on the electrodes, but you see the electrophysiology. So I think as a combination of both is good. But for our purpose, we use now calcium imaging for getting an impression how the cells look in the beginning, and it's really well adapted in our both laboratories in medicine and physics. So this is what we used before, but right now we're setting up the MIAs, because the MIAs have more power to get more detailed information about the real function of the activity, the neural activity, since it's an direct indicator for the electrophysiology. So I think we're moving now to the MIAs, to set up the MIAs and to stay with the MIA. But I think it's good to have the calcium also, just to see the cells also and to see how they look in the movies.
Okay, so if it's a short question, okay, thanks.
Very nice talk. So how reproducible is your system? Because I guess for training you need to have a very reproducible system all the time, just to get like a robust response that you have to use later for maybe implementing other treatments or whatever. So, and stem cells, one of the caveats of working with stem cells is that sometimes from diff to diff, it changes how the culture responds, and MEAs reflect that. In many papers you can see that, different differentiations, multiple lines, change the patterns of activity. So how reproducible is your system?
Okay, so right now we are not only in Barcelona, but we're also in UK having a stem cell laboratory where we work with different kind of protocols, and we are here generating cortical neurons, and there they produce different kind of like GABAergic, glutamatergic cells as well as astrocytes to play around with the ratios, combining these cells. And I think in the end, when we decide which protocols we will use, and then just using the same protocols, we should be able to reproduce the same neurons. I mean, of course, every batch is kind of different, but the final cell type should be the same if you produce the protocol in the right way. So I think it is reproducible. And I mean, the aim is also going to be this is like also the good properties, I mean, to have a neural circuit which is like trainable and adaptable to new circumstances. So I think if there's the protocol always like used in this proper way and really good adapted over time, then it should be like this, the base should be stable, this network should be stable, and then introducing different kind of stimulations. Of course, it will be different outputs of the cells, but I think the base of the network is going to be, I think, reproducible.
So every single time you add a little change, like using a different cell line, different model system, like disease cell line or something, you have to correct your deep learning method?
It's a good question. So I don't know directly, since I'm more in the biological part, to be honest. I'm more working in the stem cell part, but our partners in the computational biological science, for example, Joan, they would be able to answer this. But right now they also work in a lot of stimulations and bioimplantation parts, so they for sure would be able to implement this. So I know the stimulations they perform right now, also the codes, they use a lot of parameters, so there's a lot of talking, communication between biological part and the bioinformaticians, because these two words are kind of different from each other, to try to talk about different parameters which needs to be think about, which we need to include into the algorithm. So there's a lot of communication also happening to introduce a lot of parameters which are important in the biological part to the stimulation part. But this is the best answer I can give you in this part.
Thank you, very nice work.
Sorry, there's no more time for questions. Sorry about that. Okay, go ahead. Christina, go ahead. But please, short. If we're more efficient with astrocytes.
I mean, the astrocytes are appearing on day 30 in our differentiation protocol, and we need the astrocytes also for the survival of our neural culture. So without astrocytes, they wouldn't be able to survive for a long time. And since this chip is able, in the future, we want to keep it for a long time. For example, there are protocols to differentiate cells, neurons about 2 years. So for sure, we will need also astrocytes, which are important for the network activity and the function activity, as well as the maturity of the cells too. So yeah, we will need them too.
Thank you.
Okay, so before to flash to lunch, I have several reminds. So first, thanks all the speakers for this nice session. And also the audience for questions, discussion, and comments. So now, before to go lunch, the first remind is that we have 2 awards. The first award is for the best flash talk, that is €500, that is really good. So, and the second one is for the best poster, that is €300. So I ask, please, people that is presenting a poster to be close of this poster, because there's an evaluation committee moving around. So please be close to just explain the results in your poster. And the second remind, or the third remind, I don't know now. So it's that there's a special food for these guys that have some kind of allergies or some problems with foods. There's a specific point for these guys. So please try to find this point now in the lunch. And see you at 3 o'clock.